Hi everybody, I'm a PhD student of Hochschule. We've talked about matching cell lines for cancer subtypes. So considering the cell lines, which is a very important model system that are costly, efficient, easy to handle in laboratory conditions, and did not inflict ethical issues involving research with human or animal subjects. We feel that it's very important to identify the cell lines that are most similar to tumors. There are a lot of efforts preceded us, just to name a few. For example, selector. This work just use a sequence of pre-selected recurrent mutations to identify the subtypes and map the cell lines to bulk tumors. This might be useful. However, due to the patients or the cell lines harboring the same mutations, their frequency are very low. For the single driver events showing the left pie chart, as you can see, the frequency is very low for most of the single events. If you have the combination of the driver events showing the right panel, then the frequency is much lower. So this will greatly limit the statistical power to detect the most optimal cell lines for bulk tumors. In other cases, which are based on the independent association analysis on different data types, for example, gene expression, methylation, or mutation. So this might be useful. However, this independent analysis will result in very inconsistent results. Shown in this heat map, the rows are cell lines and map to the bulk tumors, and the columns are different data types, and the color or the number encode the confidence that a cell lines map to a tumor. So we do not need to care about how they get the score, but we only need to remember 20 means the high confidence, and 0 means the lowest confidence. So as you see, for most of the analysis, the results are very inconsistent, even though using the same data type. For the best example is MS96, that is a mutation signature, and the OGM, that is oncogenic mutation. For the two types of features, they are all derived from somatic mutation profiles. However, you see most of the cell lines, they have very different results. So we were asking ourselves, is there any way to integrate a lot of different data modalities to identify the most optimal cell lines for bulk tumors, including somatic mutation, copy number variation, and gene expression. In our settings, for the cell lines, we have for the cell lines, there are no labels, but for bulk tumors, we have the labels. So a semi-supervised VAE is perfect for solving this problem. I show here is our workflow. We first project those very heterogeneous data set, including copy number and the mutation profile onto annotated pathway network. by running a diffusion algorithm like a random work on the network, we can propagate the binary signal into a continuous matrix enriched with the network information. Therefore, we get a dense data. And then combining the network propagated data together with the gene expression profiler, we formed the two views of the MFmap input layer. In our implementation, MFmap consists of 3 components. One is encoder, another one is a decoder, and we also have a classifier. After training, once the training is finished, we are able to use the latent representation extracted by MFmap to do following analysis, such as doing the annotation, characterization, or comparison. In order to implement such a accurate model, we consider such a diagram. We thought the latent representation determine both the features X and the subtype labels Y, and using from a probabilistic perspective, we can write the evidence lower bound for unlabeled data as a general VAE, because we don't have this branch. And for the bulk tumor, we can derive an able that is a general VAE together with a loss term here. And another question you might already faced is the extracted latent representations. They are not overlapped between bulk tumors and cell lines. In order to solve this problem, we empirically found that when adding an entropy term, then this will serve as a regularization to guarantee all the features extracted in latent space overlapped between cell lines and bulk tumors. And combining the labeled loss function together with the unlabeled loss function, we get the final MFmap loss function. We first do the evaluation for the performance of MFmap, as MFmap is a semi-supervised VAE, so we hope that it can achieve both good generative and predictive performance. In order to do so, we recorded the loss term in our MFmap loss function, and then we calculated the pairwise loss term correlation. As you can see, for each pair, their correlation coefficient show in each cell, they are almost near to 1, suggesting that MFmap can achieve good generative and predictive performance simultaneously. And in more detail, because we tested this in 10 cancer types using the unseen data, 20% of the unseen data during training, and so for we used the general predictive classification performance, including the overall accuracy, weighted precision, weighted recall, weighted F1 score, and you can see the for the F1 score for all the 10 cancer types we studied, they are over 90%. Regarding the generative performance, we just calculated the input and the reconstructed genome and plot the histogram here. So as you can see, for most of the genome features, the input and the reconstructed features are highly correlated, suggesting that the MFmap is is a very good generative model. And then with the high performance of MFmap, we are able to select the best cell lines for individual tumors. So using this distance, this similarity matrix. So each rows is a cell line, and each column is a bug tumor. If you have a bug tumor here, and the green color encode the most similar cell lines. So you can also see from the label using the same color to encode, you will get the cell lines most similar to that specific bug tumor. And another usage of the MFmap is that, for example, for PA-like this subtype of glioblastoma, there are no any cell lines available now. So this will guide us in the future. Maybe we need to put more efforts to develop a cell line that are highly similar to PA-like subtype. And to explore the potential translational usage of MFmap, we just calculated the correlation between the latent representation score together with the drug sensitivity. Drug sensitivity is measured by AUC. For those who are not familiar with AUC, AUC is a measurement for drug sensitivity. A high AUC value represents resistance, and a low AUC represents sensitive. So so first we show the example lapatinib. This drug is already used to treat HER2-positive breast cancer. So in our result, we showed that this HER2-enriched subtype is more sensitive to lapatinib compared to other subtypes. Do I have time?
No.
Oh, okay.
No, you're running out of time. Just speed up, that would be fine, because very interesting, but...
Oh, yeah.
You should speed up for the last slide.
Yeah, thanks. And for glioblastoma, these two compounds are already have been discussed for treating the glioblastoma, but our results shows that for the GCIMP high subtypes, they are more resistant to the KHS101. And then we ask whether the MFmap extracted latent representations have some biological meaning. In order so I argue that the answer is yes. For example, for the GCIMP high subtype, it has a relatively low C1 score and relatively high C2 score. Similarly, another example is LGM6GBM, and so it has a higher C1 score but relatively low C2 score. This indicates that we really learned something that are highly related to cancer subtypes in glioblastoma. In order to further characterize the...
Maybe you could summarize the rest of it in a few words that would help?
Yeah. And then we correlate the pathway activities together with the latent representations. And this allows us to annotate each of the latent representation vectors. So we just make them here, and then we project the we use the bulk tumor to generate such a reference map. Show here the contour represents the density of the samples. And once the reference map generated, we are able to project our cell lines into the reference map. As you can see, the cell lines were overlapped with the bulk tumor high density regions. This is the function of the entropy. And then we are asking whether we can use our model to modeling the disease transformation. In order to do so, we consider a subtype that are not so have a relatively good survival. That is the that is the GCIMP high, and then we consider another worst case with worst survival and metastatic potential, that is mesenchymal-like, and we did a perturbation by adding a latent representation difference to the original samples, and we see the perturbed samples just overlapped with the target samples here. This is the artificial samples. And we also see the features like IDH1 mutation, ATRX mutation, or TP53 mutation, which is characterized in the GCIMP high samples, and after perturbation, their mutation score become lower. This indicates that our MFmap really good at for generative task. So really summarize what we have already shown. MFmap is a novel semi-supervised VAE. MFmap can achieve both good generative and predictive performance simultaneously. MFmap enables predicting cell line subtypes and its similarity with bulk tumors simultaneously. MFmap allows us to bridge in vitro drug screenings with clinical applications. Yeah, I think that's all.